Hello, class. A very good evening. How are you? How is everyone? How's things going in your life? So, we came for the third series, Principle of Genetics today, and we will go through a lot of exciting things in it. Many basic concepts related to genetics, um, and some of no. So let's start everything at 8 then. So we wait till that time. Um, So his voice is good so far, and you can see my screen easily on YouTube. Everything is good. Please thumbs up if everything is good, or give smiley because life is about keep smiling. That's I always say. Keep smiling. Okay, good, good, good. So where are you all from? From every all part of India, north, south, west, east, central. Uh, yes, great, great, great. Okay. Assam, Kolkata, Punjab, Kolkata, Assam. So everywhere, from everywhere. And in bachelor's or in master's? Uh, what are you studying? So you're from bachelor's mostly or master's? And what are your expectations from this workshop? What do you think that you will learn? Second year bachelor's class. Oh, there's some from class 11th also. That's great. That will be a bit advanced for you, but you will learn a lot. Masters in zoology. B Pharma, Masters, Bachelors. Okay, okay. Yes, yes, you will learn, bache. Yes, yes, this is lecture is all about to learn about genetics and gene expression. That I will try my best. So our lectures. There are two things. Somebody has signed for their project, and somebody has signed for internship letter. So both are different things. So for project, what you have to do is, okay, before I start, let's wait for another 2 minutes because I want to start when everyone is joined with us. Because there will be some new students from the that you have not joined the previous workshops. So some will be who have joined the previous workshop and joining us here also. So yeah, okay, okay. Genomics. I will share everything with you soon. Just wait for a few minutes, few 4 minutes. So it's 8 o'clock already. I will wait for 2 minutes more, then we start. So you are all over from India, doing bachelor's, master's, class 11th, pharmacy, biotechnology, zoology, bioinformatics. Some students from final semesters of bachelor's, B Pharma, zoology, from Jalandhar, from Maharashtra, Tamil Nadu. So It's Varanasi also. I want to check Varanasi once in my life. Kolkata. Oh, same here. I want to check all places, especially South India also, because I'm from Delhi. Basically, my name is your trainer, and my name is Simranjit Singh, and from basically uh, from my education point of view, after my plus two, I did my bachelor's in biotechnology from Shahid Singh College in Chandigarh, and then afterwards I went to Germany for my master's in agrobiotechnology, where I worked at the Max Planck Institute on the identification of novel endothelial specific molecules involved in the regulation of the tumor cell extravasation. Then, from my master's, I went to my doctoral studies in Germany itself at the Göttingen under the Institute of Pharmacology and Toxicology in University Medical Center, where my topic of my thesis was redox regulation of protein phosphatase 1 and ER stress regulation of connective tissue growth factor in cardiomyocytes. And beyond that, before leaving to Germany, I was lecturer in the field of Department of Biotechnology. Also, I was research assistant for more than 3 years at the University of Göttingen. And then I was also assistant professor where I was teaching principle of plant biotechnology, principle of genetics, principle of cytogenetics also in the Department of Biotechnology. And nowadays, as the COVID situation is there, so I'm online professor with Edufabrica, who have provided me such a great position to meet such awesome students from all over the world, from all over the India, and to teach you the basic things that what I have learned in my life. So so I had already delivered a 10 days workshop with them in biochemistry, molecular biotechnology, and plant biotechnology. So this is the fourth series that I'm running is now principle of genetics. And more are coming soon also. So we will be also dealing with principle of proteomics and principle of bioinformatics and principle of biochemistry for 10 days workshop. So these are also coming soon. And yeah, so that's what it's all about me, who I am. So I have lived in Germany for 7 years. If somebody has any questions, uh, so regarding to study in Germany, you can ask me anytime. I have plenty of experience for the same. I have a lot of questions coming from the previous workshop. Also, didn't reply to them, so I will try whenever I get a time. So for you also, whenever you send me questions, I will reply for sure. They are with me. Whenever I get free, I will reply to everyone. It's just a lack of time. That's it. So now comes to your uh, about projects and internship. So for projects, what you have to do is you have to write an assignment in which you make a summary of each day, 2 day summary. Sorry, 2 page summary, whatever you have learned from today's workshop, and you just write it down. You can write via pen also, or type in at the type in with the help of uh, with a word pad. So it's up to you. Yes, bache, we will teach prokaryotes regulation, transcription also. It's not so difficult. So let's see how things will work out. So there will be a objective for this workshop. So we will stick to that during our study. So this is the objective of your principle of genetics. So we will deal with the Mendel's law, types of gene actions, qualitative and quantitative traits, multiple factor hypothesis, cytoplasmic inheritance, CIB techniques, gene expressions and differential gene activations, lack of profound fine structure of gene, ultrastructure of cell organelles and their function, study of chromosome morphology, number and types, karyotype, ideogram, mitosis, meiosis, DNA structure, function, types, mode of replications, repair, RNA structure, function and types, transcription, translation, genetic code, outline of protein synthesis, linkage and estimation of linkage, and also numerical chromosomal aberrations and evolution of different crop species like cotton, wheat, reticle, brassica, and structural chromosomal aberrations. And also none and last, we deal with the mutations part, crossing over, a mechanism of crossing over with cytological proofing of. So each, I mean, it's too much, it's written here, but each session has to be done for 2 hours. It could be sometime 1 hour also. But my main focus is to finish with the syllabus here. So if we are finishing sometime early or we're finishing quite late also, so we will see. But you will have every day the recorded version of this YouTube. So don't worry. Even though if you are not present, you can watch it later at increasing the speed and then go through the lectures as per your things. So now comes the training plus internship. So with the project people who are just for the projects, who didn't pay extra for them, there is assignment you have to write. Assignments have to send to my email ID. I will share my email ID here, where you have to make a summary of each day, 2-page summary. So simranrihals99 at the rate of gmail.com. So you can send your assignments here, and they are not compulsory to send actually. But I will recommend that you please share with me so that whenever good evening, bache, whenever you are out of focus from the subject, but you have that assignment that you have done in this workshop, it will be like a bible to you. So you don't have to go through all lectures to YouTube lectures and go through again. You just have 2-page summary of each day, so that you can remember, okay, sir have explained that, sir have explained that. So it will be good for you. And about internship letter, what you have to do is you have to write a review. So topics I will share with you. So some of my students from my previous workshop, without my help actually, they even publish it in good journals also. So that was a very great task. So what I did was, so what you can do is you can make a group now. You can talk within the uh, WhatsApp group, and I will share you topics by tomorrow actually, and you can send it me. Publish bache, they are free journals actually, where you can publish. So many students have already published in IJCRT and many other places. So look for that. And then what else I was saying? Yeah, so just I will share the topics with you about the publication, and then about the review, and just make group of 5 to 6 students, and you work in a group. But many people have done their own efforts and they did it actually. So it is just about how much effort you put in. It's all about how much effort you put in. Please share me your paper once on my email ID. I will go through it and we will see how what we can do. So it is your own effort. So my LinkedIn ID is also there. So you can join me on LinkedIn also if somebody know about LinkedIn. It's by Simranjit Singh. So this is my LinkedIn group. So just send me your request. So I will show you like how many students they're sharing their papers, publications, their interactive letters, and many things. So just join me there. It's Simranjit Singh with this face. And Yeah, you can select your own topic also. That's not a problem. So that will be really appreciable if you could select by your own topic and share with other students, because working in group is always good, always good actually. Yes, it should be linked to genetics because we are studying genetics here. So the reason of purpose is that whenever you get the letter also, it will be written that you have studied the principle of genetics and you publish this paper and your certificate. But if you publish something off the topic from the genetics, that will be a mismatch. So concern for review would be to be very concerned with your what you have to work out. Okay, okay. Mannat needs someone who had never worked with someone who had worked with review. So please, Mannat, uh, have someone in the group where they have did it before, because it's all new experience. So students will really help each other and make it done. So it will be done. Don't worry. Many students publish it actually. So okay, I will share some of the assignments. So how you have to do it. So see, so many reviews and assignments I have got. And very nice reviews also. So like this person sent me everything in 10 days format, each day summary in one folder. So I was teaching about cell, so some diagrams, how it works, some purification of isolation of DNA I've taught. So in just 2 page, all the topics were covered within that workshop. So within this assignment. So that's how you have to deal with it. So, and one of my students who have done nice review, like this one was very nice review somebody wrote. You can see very nicely done with the heading, public authors, then from the place they were, and then that was the abstract is was there. So then after abstract there is the introduction. This is the format of review. Then preparation and properties of your things that you're working for. Then subheadings as much as possible. So these are subheadings. Then at the end, conclusion. Then there's author's contributions, references, very nicely done, and then that's it. So this is how a nice example of a review, I would say. So topics I will share tomorrow, uh, but you can also choose your own. But at the moment, I will share uh, a review format, how to write the review. My email address. That's it. I hope everything is clear, because most of the students ask me again and again, sir, what have we have to do? What is the things we have to make? So when is the last date? So for the review, the last date would be 5 days, uh, uh, 5 days from the workshop end. If our workshop ends on Today is 16, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 25th if it is finished. So 1, 2, 3, 4, 5, 4. So by the end of month, you have to send me the review. So you have plenty of time. You don't have to anyone give to NEET now. You don't have any exams now, I guess. So you have plenty of time to do so. So today is 16th, then from 16th till 30th, you have so many days. So just be careful. So you have 15 days or so. So just take your time, think about it, and do it then. Make it happen. So on that note, I hope things are clear. So today will be a light day, a little bit introduction. I don't want to keep it heavy because I have seen some complaints that the people when I start too much. So I want to keep it light, halka fulka, so that we start gradually with the large parts, with the more things in it. So this lectures will be more subjective actually. In the earlier lectures, I was giving a lot of videos and a lot of interactive things, but in this case, these will be a lot of lectures. It will be like a college format lectures, and because this syllabus that I'm teaching you is from the Indian Agriculture Research Institute. So from the basically a confined and very exact syllabus for the principle of genetics. So, and I will cover each topic that I have discussed with you today. So, on that note, let's start. So, I will cover mainly two topics for today, that is history of early concept of inheritance and Mendel's law. That these two topics will be covered today only. So, in principle of genetics, the first topic is our definition, history, and early concept of inheritance. With the help of Edufabrica, I'm presenting you this lecture as your trainer, and I hope we had a good time and good sessions in upcoming days. So let's start. So this is your syllabus looks like that I have discussed in early part. So it will be divided into two parts, that is unit 1 and unit 2. So your all parts will be covered from your unit 1 to unit 2 in this case. It's a lot of lectures, so it's a uh, one semester presentations are there, but we will try to cover them in 10 days. And so be with me during this time, and I hope we cover each topic with great concept and the basic principle behind each topic that has been there written. Okay. So, principle of genetics. So, in this topic, we deal with definition, history, and early concept of inheritance. So, if we see from the day-to-day life, from the mango tree, if we put him in the soil to grow, we will mostly get a mango plant out of it. And from the dogs which give birth to puppies, they will give to young ones of puppies only, not any other animal. And humans also give birth to the human beings only. So this tendency that we can see that whatever the the genus is, the species is, their offspring to inherit parental character, whatever the parental character they are inheriting, is termed as heredity. And the study of science of heredity and the reason governing the variation between the parents and the offspring is called genetics. So, and so this is the heredity, that inheriting the characters to the offspring. And to study this science of heredity and reason the governing behind the variation between parents and the offspring is called genetics. So in the history, um and students, let me just, before we go ahead, I want to just check with the, because sometimes things are not good happening. So students, is my voice and the sound is going fine with the lecture, so there is no lagging is happening and no extra sound is coming from outside? If all is good, please confirm with me so that I can start with the lecture. No, sir. Everything is fine. Okay, that sounds great. So continue. So first in the history, Augustin Friar Gregor Johann Mendel, that's the full name of Mendel, who is known as the father of genetics. He worked on the work of pea plant and published in 1866, but was not recognized during that time, and it called as work to be called as Mendelian inheritance. Then in 1900, rediscover of Mendel work happened with the help of Hugo de Varis, Karl Correns, and Erich von Teschmark. So these people rediscovered his work, and basics of principle of Mendelian genetics have been widely applied to various other organisms also in 1915, such as your Drosophila melanogaster, that is fruit fly. Then Thomas Hunt Morgan and his fellow Drosophilist and genetist they developed Mendelian model, which was widely accepted by 1925. Then in the 1940s and 50s, experiments were pointed to DNA as a portion of chromosomes that held genes. And with the discovery of double helical structure of DNA in the 1953 marked the transition to the era of molecular genetics. So some early concept of inheritance. First, let's start from the Aristotle and spontaneous generation. So what does Aristotle says that all the things uh, that first he recorded conclusions based on the roots to the life. He saw things being arised in one or three ways. That is, sexual reproduction, asexual reproduction, or by non-living matter. So it was observable that aphids, that is the fruit flies, they were arising from the dew on the plants. So like all the waste matter of the plants, fleas from the putrid matter, and mice from the dirty hay. And these beliefs remain unchallenged for more than 2000 years. So he was the first one who tell that from the dirt, actually from the dead matter, life arises, and that remains for 2000 like that. So in this case, if there's a dead matter is there, so on top, food flies is coming from outside on the dirt, but he says they are arising directly from the plant, but that was not the happening. So that was but that was still accepted for 2000 years also. Then he ready challenged this concept by spontaneous generation. So Reddy challenged the doctrine of spontaneous generation. Reddy question was simple: where do these maggots came from? So, according to spontaneous generation, one could conclude that maggots come from the rotting food. So Reddy hypothesizes that maggots coming from the flies designed on the experiments. Actually, it was not the maggots that coming from the rotting food, but was coming from the flies that were coming on the top of your waste food actually. So he what he did is he Francisco Reddy experiment in late in 1600s. What he did is he had a flask and he opened with the help of, with the meat in it. So he left the jar open in the first case. So the flies came and they had the maggots on top. So and then in the second case, flask was sealed, so no flies came. But then there he covered with the gauze with a sheet of thin cloth paper, and then he saw that flies were coming and the dead matter, the maggots, they were being produced on the top of the sheet. So this proves that it was not happening because of the dead matter, but it was happening because of the flies outside. So jar 1 was left open. He observed flies lying on the meat, and then eventually development of maggots was happening. And then in the part 2, a jar covered with the netting, flies laid their eggs on the netting and maggots soon appeared on it. Then flies were not attached to this jar and no maggots developed on the meat. So this seems to be clear demonstration of life giving rise to life. Yet it took another 200 years for people to accept spontaneous generation as a fallacy. So it still take them to 200 years to accept spontaneous generation as a fallacy case also. So Anthony von Leeuwenhoek, then he came with a thing called animalcules. So Leeuwenhoek was a Dutch cloth merchant. Due to his frequent movement of for in the ships, he used lot of lenses. So he employed lenses to see his clothes, but sometimes in his ship, he was curious about microscopy also with different organisms. So he discovered various microorganisms so tiny that they were invisible to the naked eye. So he called those microorganisms at that time as animalcules, which were first to describe many microbes and microscopic structures, including bacteria, protozoans, and human cells. Then early concept of these were the animalcules that what he discovered. That was his paper. As you can see, animalcules written by Leeuwenhoek. So his piece of work. Whatever he saw, he just draw them like this. Then comes the in the early concept of inheritance, Louis Pasteur. So he was a French scientist who made great contribution to our understanding of microbiology, and for whom the process of pasteurization is named after his great work in pasteurization. So he brought the light of strong evidence of microbes arises from the other microbes, not spontaneously. So they were not growing spontaneously from itself, but they were from the other microbes they were growing it. So for that, he created experiment called Pasteur-Swanneck's flask experiment, where he created a glass flask with unusual long leg, long neck, thin neck that pointed towards downward. So like this one, he created a long swan neck shaped, and then swan neck flasks were allowed into the air container but did not allow particles from the air to drift down the body of the flask. So they were like in this case, this infusion was boiled for a long time to kill all the microorganisms in the present. So there is a medium, so they kill all the microorganisms in that flask and let the air go in, but microorganisms were trapped in this swan neck shape. So after several days, no microorganisms were appeared in this case, but then the flask was tipped to allow the trapped microorganism to enter the infusion. So it was tilted a bit so that the microorganisms that were staying there in this case, they could come inside. So microorganisms quickly multiplied in the infusion. When we did that, they started to multiply in it. So that was a Pasteur experiment to show that the microorganisms were coming present from the air, not from the within the medium itself. So this also and the spontaneous generation theory. So after boiling his nutrient broths, Posture found that these swan neck containers would remain free of microbes until he either broke the neck of the flask, allowing particles from the air to drift in, or until the tilted flask so that the liquid came in contact with the dust that had accumulated at the opening of the flask. It was these careful control experiment of Pasteur that finally put the rest debate over spontaneous generation. So that's it. Then comes the next part, uh, that each organ of an individual produces very small, almost invisible identical copies of itself called gamules, that is pengenes. So these gamules from various parts collected into bloodstream of animals. So he says that each individual is producing self-identical copies called gamules or pengenes, and they are collected into the bloodstream of animal, and the blood transport the gamules into the reproductive organ, and which produce gametes further. So somehow he was right, but not fully correctly. and male and female gametes unite to form zygotes. So when these give rise to new organism, the gametes of different part of body give rise to same kind of organ, tissue and cell, which produce them in the parents. So this is your body, which are producing in our blood cells gametes, which get in touch with the egg, and then hence development of embryo is happening. So then comes the Lamarckism. That's a famous scientist who produced Lamarck theory, a French biologist from 1774 to 1829. He considered the inheritance of acquired character to be the most important. If not, the evolutionary changes are the responsible for it. So according to the urgent need, use and disuse of organs is happening. So whatever the use and disuse of organs are there, then there will be modification happening to that organ that will be transmitted to the offspring. So whether we are using some organ or not, according to that, that inherited character will be transferred to the offsprings and will be lead to the, yeah, change in the organ. So, for example, in this case, famous example by Lamarck giraffe. So, originally it has a short-necked ancestor, and then it keeps stretching neck to reach the leaves higher up on tree, and keep stretching until it's get to its food, and then at the end progressively it's getting longer and longer and longer. So that's how your Lamarckism theory works here somehow, I have to say. So then further, plasm theory, August Weismann. So he brought the germplasm theory by August Weismann. That is explains the body of individual consists of two distinct tissues. So he brought further better version of Darwinism. That what he was saying, that somatoplasm consists of all body tissues which do not contribute to the sexual reproduction, but there are germplasm on the other hand produce gametes that are basis of heredity. So he brought somatoplasm and germplasm, two new terms. So it is only applied to animals and plants in which distinguish between soma and germ can be made. Weismann famous experiments of cutting tail of 22 generations of mice and observing that the progeny still had tail of normal length. So tail was not getting removed or getting bigger or smaller. This proved that spermatoplasm is not responsible for the transmission of character. So in this case, because that was not uh, the size was still the same, and that was the reason spermatoplasm was not the reason behind the transfer of inherited characters to the different generations. So at the end comes your Mendelian concept of heredity. He brought the law of inheritance, which was derived by the Gregor Mendel, a 19th century monk. He did lot of experiment with Pisum sativum, the pea plant. Then he worked between 1856 to 1863 and cultivated around 29,000 pea plants. So he was a monk also actually in the church, but also working on these on the pea plants as his hobby on backside. So at the first his work was not characterized, but later on Mendel's law of heredity or Mendelian inheritance was famously accepted. So he described his paper in two papers: Experiment on Plant Hybridization, that he read to the Natural History Society of Brno, and which was published in 1866. So, a little bit about Mendelian concept of heredity, that he also measured only absolute characteristics such as color, shape, position of the offsprings rather than the quantitative characteristics. He expressed his results numerically, subjected them to statistical analysis. His method of data analysis and his large sample gave credibility to his data. Finally, he performed many test crosses to reveal the presence and proportion of the recessive characters. Without his careful attention to procedure and detail, Mendel's work could not have been the impact it made on the world of genetics. So here's our Mendel doing his work. So that was his work. In it is showing the 7 different characters. That is height, seed shape, seed color, seed coat, pod shape, pod color, flower position, and further there is recessive traits and dominative traits. We will go through each of them also. What is dominant and recessive trait? A dominant trait is the one which is present mostly because of the dominant alleles, that is tall, round, yellow, green, inflated, green axial, whereas the recessive traits are the ones which do not used to be present because of other recessive allele present. They highly not to be present. So it's like short, wrinkled, green, white, constricted, yellow, terminal, respectively. So on that note, our history is done, very short and crisp. So to go next, let's take a 10 minutes break, and then we see us with the next lecture, that is principle of genetics, Mendel's law. So today is a light day. I did not want to make it so big fast for you, but from tomorrow we will start the main things. So I will share some of the things here, Mendel's law. In meantime, let's meet at it's 35 or 37, at 47 or so. Let's meet in 10 minutes then. Mendel's law of inheritance. So, class, I'm back. Let's continue with the Mendel's law of inheritance. So, please come back, everyone. So, Mendel discovered that by crossing white flower with purple flower, the result was not a hybrid spring. So, whenever he was crossing purple with the white, what he was emerging that most of the time it was purple flower. He then conceived the idea of heredity units, which he called factors. So that was the reason that the things that were transferring from one species to another was something called as factors. One which is a recessive factor and other is a dominant, and he called them one as recessive and one as dominant. Later on, these factors were called as genes present in the ordinary body, and each member of pair becomes part of the separate sex cell. So the dominant gene, such as the purple color in the Mendel plants, will hide the recessive gene of the white color. So here you can see we remove the stamens from the purple flower from the carpel, and we transferred the pollen from the stamens of the white flower to the purple of the carpel of the purple flower. So we transfer from here from the white one to the purple one, and then we pollinate them into pods. Then plants were seeded. So this is your parental generation, and then examine these offspring that all springs were in purple. So the F1 generation that he obtained was in the ratio of 3 is to 1. He correctly theorized that the different genes that were present, they were capital A, small a, small a, capital S, small a. So this is what I'm teaching you is a plus 2 genetics today. So just to refresh up us that before we go ahead, we should be aware of all these things also. These all things are in your plus 2 genetics. So I'm teaching sometimes to NEET students for plus 2. So I have seen that that's similar. There's nothing much change, and you're also studying the same thing at the bachelor's or even a master's level. So these things keep on repeating. Um, but your task is to keep them fresh up with yourself. So in this case, the capital letter represents the dominant factor and lowercase represent the recessive factor. So capital A and small a, capital A and small a, like this. So in the history, we see Mendel stated that each individual has two factors for each trait, one from each parent. The two factors may or may not contain the same information. If the two factors are identical, the individual is called homozygous or for the two trait. If the two factors have different information, the individual has heterozygous. If they are different, then it is a heterozygous. If the information is identical, then it's a homozygous. So alternative forms of factors are called alleles. And the genotype of an individual is made up of many alleles it possesses. An individual physical appearance or phenotype is known as per the environment. It is determined by its allele as well as its by environment. An individual possessing two alleles for each trait, one allele is given by the female parent and other by the male parent, and then they are passed on to the individual produces mature gametes like egg and sperm. So in a heterozygous individuals, the only allele that is expressed is a dominant, and the recessive allele is present but its expression is hidden, like capital A and small a. So your capital A will be present and your small a will be not present in that case. So that was something about history of Mendel's law. Now comes the Mendel's law of inheritance. So he has postulated three laws, which are now called by his Mendel's law of heredity. So these are uh, law of dominance and recessive, law of segregation, law of independent assortment. So law of dominance states that definition states that when two homozygous individuals with one or more sets of contrasting characters are crossed, the characters that appear in the F1 hybrid are dominant characters and those do not appear in F1 are recessive characters. So whenever two homozygous individuals with one or more set of contrasting characters are crossed, and the characters that appear in the F1 hybrid are dominant characters, and those do not appear in the F1 are recessive characters. So only the dominant character will transfer and the recessive character will not transfer in the law of dominance. So this is the explanation of law of dominance. So here you can see there is a cross between capital B and small b with the pistil, capital B and small b with the pollen. So this is your male part, this is your female part. And in the end you have capital B, capital B, capital B, small b, capital B, small b, small b, small b, which is recessive, which is a white color. But a purple color is capital B, capital B, capital B, small b, and capital B and small b. So you got a ratio of, phenotypic ratio of 3:1. That is 3 are your purple and one is white. So in this case, dominant and recessive of genes can be explained on the basis of enzymatic function of the gene. Dominant genes are synthetic active polypeptides that form functional genes, whereas recessive genes code for the incomplete or non-functional polypeptides. So that's why you see dominant character to be present because of the synthesizing and active polypeptide or proteins that form a functional enzymes, whereas the recessive genes code for the incomplete or non-functional polypeptides. Therefore, the dominant genes produce a specific phenotype while the recessive gene fail to do so. So that's why the phenotype is always the dominant are presenting, whereas your phenotype of recessives, they are not presenting anything. So in this heterozygous condition, also the dominant gene is able to express itself so that the heterozygous and homozygous individuals have similar phenotype. So law of dominance, what is it says? If there are two alleles coding for the same trait, one is dominant, it will show up in the organism while others won't. So what are the importance of this law of dominance are? There are many importance. So first importance is practical importance, that as the harmful recessive characters are masked by the normal dominant characters in the hybrids. So dominant characters might mask the harmful recessive characters in the hybrids. In human beings, a form of idiocy, diabetes, hemophilia are recessive characters. A person hybrid of all these characteristics appears perfect normal. The harmful recessive genes can exist for several generations without expressing themselves. Exception to law of dominance is the incomplete dominance. And after Mendel's several cases, uh, the scientists identify that F1 hybrid exhibited a blending of characters of two parents. So they were blending. We will discuss about it, about incomplete dominance also in upcoming sessions. So for incomplete dominance, that means also it's here only that there's a, in 4 o'clock plant Mirabilis jalapa, plant with the red flower were crossed with the plant with the white flower. Capital R, small r. So we should see in normal case that your flower should be all red, but what we see that there was a hybrid came out which was bearing a pink color, and this is called as incomplete dominance. So when these F1 plant with the pink flowers are self-pollinated, so they were self-pollinated these F1 plants, then they produced a new ratio, that is red is to pink is to white into 1 is to 2 is to 1 for the F2 generation. So that was quite surprising for all the scientists that why this was happening, and this could not be explained by the law of dominance. Uh, that was some exceptions to that. Further, law of segregation, that is purity of gametes. Mendel summarized his finding into two laws, that is law of segregation and law of independent assortment. So law of segregation says the purity of gametes, that your gametes states that individual produce gametes and these copies of genes separates so that each gamete receives only one copy. So like in this case you have capital R and capital R small r. From them single gamete is coming out, capital R and from this small r, and they together making capital R small r, and then you are making a cross of self. Then you are producing round into wrinkled 3:1 ratio by crossing them. So law of segregation states that with a pair of contrasting factors or genes are brought together in heterozygotes. So they are pair of contrasting characters, capital R and small r, round and wrinkled. Bring it together without being contaminated. And when gametes form the hybrid, the two separate out from each other, and only one enters each gamete. So one is separated, and mainly the one is entered into the gametes, like this in this case. So this is also another example of law of segregation, that there's a purple and white flower. You make a cross with the F1 generation. All plants have purple flowers. So fertilization among F1 plants, F1 multiplied by F1. F2 generation is a 3 by 4th plant, and F1 and the F2 generation has also 1 by 4th of your white flower plant. So this is all happening because of the law of segregation. So how this is happening? So you have genetic makeup of your plants with a capital P, small p, small p. From this, your gamete have only one capital P and small p, and you make a hybrid, capital P, small p, and you take half of the gametes then, and then you make a cross again. Then in the cross, you see three purple and one white, and genotypic ratio is 1:2:1. So that's how the mainly the gametes are playing the role in this purity of gametes in the law of segregation. Then comes the third law, that is law of independent assortment, that is also known as inheritance law, which states that the alleles of different genes assort independently of one another during gamete formation. So while Mendel experiment with mixing one trait always resulted in a 3:1 ratio between dominant and recessive character, uh, and his experiments mixing two traits showed in a dihybrid cross, whenever he do the dihybrid cross, a different ratio was coming, that is a 9:3:3:1, and each of the two genes in that case independently inherited with the ratio of 3:1. So Mendel concluded that different traits are inherited independently of each other. So it doesn't matter how many hybrids you do, but these are inherited independently of each other. There is no relationship between them. So it's like cat color is inherited and tail length. So they are not interdependent with each other. They are independently inherited. This is actually true for all the genes that are not linked to each other, and this is also happening in our humans also. So like this law of independent assortment. So here we can see this is a male gametes, female gametes with round and yellow. Round is a dominant, and yellow color is also dominant, and these are heterozygous RY, then recessive RY, and RY capital Y. So then they are crossed together, so they are 1/4 of each, and you saw the ratio to be present as 9:3:3:1. So here the yellow ones are the round yellow, the green ones are the ground green, and there were also wrinkled yellow, and one was present as a wrinkle and green, the recessive one. So that's how your things works out in this case. So they are independently assorted. So round and yellow, they are not dependent on each other. So they are both different characteristics, both different alleles, and they are working separately from each other. So that was something about that point of view. Now we see same thing. This is the cytological experiments they are explaining us. This is more advanced version with another example actually. So where they are using a plant yellow and round. So they are explaining the same thing what we have discovered there in detailed manner. So there are Mendelian deviations. Um, so they are the one. There are three types of Mendelian deviations. That is, which were not uh, as per the act of the Mendelian's law. So they are incomplete dominance, codominance, and lethal genes. So incomplete dominance we have already discussed in the case of Mirabilis yellapa, where there's a cross between red and white flower was done and produced plants with the intermediate flower color, pink color in F1 generation, and modified ones in the ratio of red is to pink is to white in F2 generation. So red flower multiplied by white flower, you got a pink flower in the F1 generation, and then you again cross it by itself. Then you have a red is to 1 is to 2 is to 1 in red, pink, and white respectively. So this is your incomplete dominance. This is explained with better diagram. Then it's a codominance. In case of codominance, both alleles express their phenotype in the heterozygous greater than the intermediate one. So let's see, the example is of human blood group. So AB is a blood group of human, and the people who have AB blood group heterozygous exhibiting phenotype for both IA and IB genes. So it will be exhibiting both for IA and IB. So in other words, heterozygous for codominant alleles for phenotypically similar to both parental types. So so they were quite they were like belonging to the parent also and having the phenotype of both genes also. So the main difference between codominance and incomplete dominance is lies in which the genes act. So in case of codominance, both alleles are active. In other incomplete dominance, we saw that there is hybrid of both, but in codominance, both genes are active, while in the case of incompletes, both have intermediate effect. So, like in this case, you have codominance of capital D, capital T, capital T, and small t, and small t, small t. So, this is your tall, this is your dwarf, and this is the mixture of both. But in this case, in codominance, we see the both effects together. So, like this, and we have both effects of this in the codominance. And in the lethal gene, last not the least, what we saw here is lethal genes are the one which cause the death of its carrier. So when in homozygous condition is called lethal gene, Mendel's findings were based on the equal survival of all genotypes. So normal segregation was 3:1 modified into 2:1 ratio. So like in this case, you have your yellow, and this is also yellow. They have the we did the meiosis, capital Y, capital Y, small y, and gametes are capital Y, capital Y, small y, and you did the fertilization. And the F1 generation seems to be production. What you produced is dead, yellow, and non-yellow. So the capital Y genes, they were mice die, whereas the half of the progeny they remain yellow and non-yellow. And why is it so? Why was it happening? Because your capital Y gene was lethal gene. That was this came to be happen. And if you have a lethal gene in your species, it will leads to death of your mice. And but if you have recessive gene present, it will make an effect and give rise to a recessive gene with the non-yellow color and with the dominant character with the small y to be yellow. So like this, you have different progeny. So in this case, lethal gene have been reported in both animals as well as in plants. In mouse, like we have discussed, yellow color is dominant over gray. When cross is made between yellow and gray ratio, 1:1 yellow and gray mice were observed. They indicated that yellow mice are always heterozygous, whereas the yellow homozygous is never born because of homozygous lethality. Such genes were not observed by Mendel. He always got a ratio of 3:1 for in F2 for single gene character. So lethal gene can be recessive in the aforementioned mouse experiments. Lethal genes could be also be dominant, conditional, semi-lethal, synthetic, depending upon your genes included. So that was about so far exceptions to the Mendelian's law. Now comes the third part, that is your monohybrid cross. Uh, so cross is made between two breeding parents differ for the single trait producing an F1 generation. So in case we have a homozygous green, homozygous yellow, and we intercross to produce an F2 generation. So you make a capital, you take law of segregation, you take the gametes, you make a cross, so you have F1 generation, and all are green again. Gametes are all are green, and then you make a further cross, then you have a generation of 3 green and 1 yellow. So like a normal case, 3 greens and 1 yellow, and this is a Punnett square table. So this you might know the Punnett square. You put GG here, and you just cross them and write it like this way. So that was monohybrid cross. Dihybrid cross is where you take two characteristics, like in this case tall and dwarf, and green and yellow, uh, as a dominant and recessive characteristics respectively. So pure breeding parents can be crossed to produce a dihybrid, meaning that two genes affecting the trait as a heterozygous as a F1 generation. So this is the cross that you can see. This is a bit shady. So you have a capital G and small capital T with the cross with a small g and small t. So you make a cross with the F1 generation. You have capital G, capital T small t, capital G, capital T small t. So you have both heterozygous now after the first cross. Then you take the gametes, that is GT, GT small gt, then capital G small t, and then small g capital T, then small g capital T, like this. You have four gametes out of them. Then you write them on this side, and then you have a ratio of like this: 9 is to 3 is to 3 is to 1. No, I think, yeah, 9 is to 3 is to 3 is to 1. And then there is a term called backcross, where you backcross with the parents, with the parent generation, to achieve the offspring with the genetic identity of the close to the parents. Whereas a test cross is a statement where alleles are dominant over the others. The phenotype of organism does not always reflect genotype. A recessive phenotype is only expressed with the organism is homozygous recessive. A pea plant with a green pod may be either homozygous dominant or heterozygous dominant. So this is done to determine that whether the phenotype that you're crossing, is it a homozygous dominant or is it a heterozygous. So you just do a test cross for that. So example, a dominant phenotype, we don't know whether it's a capital P, small p. So we don't know what does it look like, but we know the recessive phenotype we have. So if we have capital P, then all offsprings will be the capital P, like this. All will be purple. But if one is P, one is small, then that means the cross has been shown that half our offsprings are purple, half are white. So that means our offspring is one capital P and small p. So this is the test cross you checked by doing the experiments. You can tell us about the unknown genotype with the help of test cross. So that's the end of today's session. Not so hefty, crisp and small, and I will try to not to make much more complicated things. So I want to keep it simple as it is, and then we go through the advanced versions also slowly and slowly. So I will share you today's task also, today's things, and will share with you the reviews tomorrow. And for tomorrow, we will be dealing with the uh, types of gene actions and qualitative and quantitative trait. So we will be dealing with this part. So today is 16th. So yeah, so we continue like this in this month, and we see us your review soon. And yeah, okay. Uh, so any questions so far? So please, students who have worked with review anything in your life before, get make some group in the WhatsApp group, and start talking with each other, and decide your topics if somebody wants to do it on your own. Otherwise, I will share their topics with you also. On that note, I would like to say good night. Take care. I will share you the each day presentations with you also, and also share with the objective of our workshops, so that we are on track with the things and we are doing things on time. Uh, Gauri, just ask around the students. Somebody will tell you for sure. So I have to go ahead now. Thank you so much. Wish you all good night. And remember one thing: If you have very important thing to do, only then leave from your house. Otherwise, don't leave. I have lot of my. I always tell to my students before leaving and start of my lecture, stay safe as much as possible because this corona is really. I have seen people getting through it, and it's not likely to be taken easy or very simple thing. So as much as group you want, you can make five, six should be the maximum. Yeah. Okay then. Good night. Take care. See you tomorrow. Same time, same place. Bye.